Dear students, in this session we are going to discuss about bacteriophages, their morphology, different morphological types, and the mode of reproduction or replication, that is lytic and lysogenic cycles of phage reproduction also will be discussed. So bacteriophages or simply phages, they are viruses that infect bacteria. They were discovered independently by Frederick Tourt and Felix de Herel. It was de Herel who coined the term bacteriophage. Bacteriophage means bacteria eater. Phage means to eat. They were named so since they possess bactericidal activity, that is, they kill bacteria. In nature, phages occur in close association with bacterial communities. They are present in fecal matter, sewage, other natural sources of mixed bacterial growth, etc. And thousands of varieties of phages exist. And each phage is specific towards specific bacteria. They possess host specificity. They may infect only one type or a few types of bacteria or archaebacteria. Phages possess high host specificity. Coming to the morphology of a bacteriophage. A bacteriophage consists of a nucleic acid surrounded by a protein capsid. This image shows a typical bacteriophage, that is a coliphage that infects the bacterium E. coli. In a typical bacteriophage, it will have a head and a tail. So in a head, it consists of a tightly packed core of a nucleic acid, either DNA or RNA, which is surrounded by a protein coat or capsid. The size of the head varies in different phages. Its size generally ranges from 28 to 100 nanometer. The nucleic acid in a phage may be either DNA or RNA, and it may be either single-stranded or double-stranded. The capsid is made up of several subunits known as capsomeres. The capsomeres in turn are made up of, or it consists of a number of protein subunits known as protomers. So, the protomers combine together to form capsomeres, and the capsomeres build up a capsid. Next is a tail. The tail is composed of a central hollow core or a tube. This central hollow core or a tube, it is surrounded by a contractile sheath. Most of the phages will have this contractile sheath, but some phages does not have contractile sheath. And then there is a terminal base plate on the end of the tail, and on this base plate, on this hexagonal base plate, several tail fibers will be attached. Some phages will have prongs or tail pins also attached onto the lower end of this base plate. So the tail composed of a hollow core or a tube which is surrounded by a contractile sheath. There will be terminal base plate and there will be tail fibers and tail pins or prongs. These tail fibers or prongs help the phage to bind to the specific receptor sites on the bacterial surface during phage replication. There are three basic structural forms of bacteriophages. They are an icosahedral head with a tail. Second one is an icosahedral head without a tail. And the third one is a filamentous form. So, examples for phages having icosahedral or cubical symmetry are 5174, MS2, etc. Phages T2, T4, T6, these are coliphages. They are examples for the phages having icosahedral head with a tail or a binal symmetry. Filamentous phages are having a helical symmetry, and examples are M13 and FG. We can see certain examples for these different structural symmetries of phages. We can see 5174, FD, M13, T2, T4, T3. These are certain examples of bacteriophages showing these three different kinds of structural symmetries: helical, icosahedral, and binal symmetries. Bacteriophages are grouped into different morphological types. Based upon their morphology, they are classified into type A, B, C, D, E, and F, and type G, which is pleomorphic. We can see the representative images of bacteriophages, different types of bacteriophages. Type A is having a hexagonal head, a rigid tail with a contractile sheath, and it will also have tail fibers. The genetic material of type A bacteriophages is double-stranded DNA, and examples for type A phages are T2, T4, T6 phages. They are polyphages. Type B bacteriophages will have a hexagonal head. Their tail is flexible. They are having a flexible tail. There will not be any contractile sheath, and they may or may not have tail fibers. Genetic material is double-stranded DNA. Examples are phage T1 and phage T5. Type C, they have a hexagonal head. Their tail will be shorter than their head. They have a shorter tail, no contractile sheath, and they may or may not have tail fibers. Their nucleic acid is double-stranded DNA, and examples are phage T3 and T7. Type D, these phages will have the head only, they do not have any tail. The head is made up of large capsomeres. Their nucleic acid is single-stranded DNA. Examples are S13 and 5174. Type E bacteriophages will have a head. They do not have a tail. Here the head is made up of small capsomeres. Their genetic material is single-stranded RNA, and phage F2 and MS2 are examples. Type F, they are filamentous or helical phages. Their genetic material is single-stranded DNA, and examples are FD and F1. Pleomorphic phages, they do not have any capsid. Their genetic material will be double-stranded DNA, and example is MVL2. Now we are going to discuss about the life cycle or replication of bacteriophages. During infection, a phage gets attached to a bacterium, to a specific bacterium, and inserts its genetic material into the bacterial cell. After this, the phage usually follows one of two life cycles. Based upon their life cycle, phages are classified into two. They are lytic or virulent phages and lysogenic or temperate phages. The lytic phage or virulent phage take over the machinery of the cell. The metabolic machinery of the cell will be controlled by the phage nucleic acid to make phage components. Then they destroy or lyse the bacterial cell, releasing new phage particles. While in the lysogenic cycle or the lysogenic phages or temperate phages incorporate their nucleic acid into the host bacterial cell chromosome and replicate along with the bacterial cell as a unit without destroying it. So the bacterial cell remain healthy even if it is having the phage chromosome, phage DNA incorporated into its chromosome. Under certain conditions, the lysogenic phages get induced and then they follow a lytic cycle, lyse the cell and then release the progeny phages. So there are two types of bacteriophages, a lytic phage and a lysogenic phage. Now we will discuss lytic cycle. Lytic cycle is a mode of reproduction by lytic phages or virulent phages. Example of a virulent phage is T2, T4, T6 phages of E. coli. During lytic cycle, the virulent phage replicate through the following stages. The various stages are adsorption, penetration, transcription, assembly and maturation, and finally the release of progeny phage particles. So coming to the first stage in the lytic cycle, that is adsorption. Here the phage particles attach to virus-specific receptors on the host cell by their tail. This adsorption is a specific process and depends on the presence of complementary chemical groups. The complementary chemical groups present on the receptor sites on the bacterial surface and on the terminal base plate of the bacteriophage will interact specifically and it will lead to the adsorption. The initial adsorption of phage to the receptor is reversible since only tips of the tail fibers are attached to the bacterial cell surface. After this, the tail pins will attach to the bacterial surface and the adsorption becomes irreversible. This adsorption is a very rapid process and it will be complete within minutes under optimum conditions. Any component on the bacterial cell surface can serve as a receptor for phages. The bacterial receptor may be part of lipopolysaccharide on cell wall, or it may be flagella, pili, membrane, or cell wall carbohydrate or proteins. As we said before, phages are host specific. They possess a high level of host specificity. An E. coli phage cannot infect a salmonella or any other bacteria. Some phages are even specific to strains of bacteria. This host specificity of phage is determined at the level of adsorption, that is the first stage of phage replication. Adsorption depends on the interaction of complementary chemical groups from the receptor site on the bacterial surface and the terminal base plate of the bacteriophage, and this chemical interaction, specific chemical interaction, is the basis for host specificity of bacteriophages. The second stage during the lytic cycle of a bacteriophage replication is penetration. After adsorption, most phages inject their nucleic acid into the bacterial cytoplasm and leave their protein capsid outside. This is similar to injection through a syringe. This stage is the penetration. During penetration, the tail fibers will attach firmly to the bacterial cell, and it will firmly attach the phage plate to the bacterial cell wall. Then the contractile tail sheath will contract. The contraction of the contractile sheath will force the hollow interior tail tube into the bacterial cell wall. Then the phage DNA will pass through the tail tube into the bacterial cell. The empty phage head and tail will remain outside the bacterium. Penetration is facilitated or improved by the presence of lysozyme on phage tail. Lysozyme will cause localized digestion of bacterial cell wall surfaces. Some phages, such as T1 and T5 phages, they do not have the contractile sheath, so they inject their nucleic acid through adhesion sites between the inner and outer membranes of bacterial cell wall. Filamentous phages and rod-shaped bacteriophages, they enter directly into the bacterial cell and then release the DNA into the bacterial cytoplasm. The third stage in lytic cycle is a transcription, that is synthesis of phage nucleic acids and proteins. The synthesis of phage components occur immediately after penetration of the phage nucleic acid. It occurs through several stages, that is the formation of immediate early genes, delayed early and late gene products. Immediate early phage genes are transcribed by bacterial RNA polymerase, since that at that time the phage does not have any enzymes, it only has its genome. So the immediate early phage genes are transcribed by bacterial RNA polymerases. Examples for the immediate early phage gene products are nucleases that will break down the host DNA, and these breakdown products will be used for phage nucleic acid synthesis, and also a few other enzymes will be synthesized which alter the bacterial RNA polymerase, so that the affinity of the bacterial RNA polymerase will be altered and it will work more for the bacteriophage. So from this moment onwards, the bacteriophage genome will control the bacterial metabolic machinery towards the synthesis of bacteriophage components and their nucleic acid replication. Next is the delayed early phage gene transcription. The products include enzymes to produce phage constituents for producing phage head, tail, etc. Then phage polymerase, ligase and RNA polymerase for nucleic acid replication, and further transcription will also be made during this delayed early phage gene transcription. Then the late gene products include structural components for new phage particles. The heads, tails, fibers, etc. are formed. So the enzymes formed during the previous stages will make the protein subunits for head, tail, etc. during this late stage. And a phage lysozyme will also be produced to lyse the bacterial cell for releasing mature phage particles during the later stages of lytic cycle. Now comes the assembly and maturation stage. This phase occurs after the synthesis of structural proteins and nucleic acids. The phage components formed during the transcription phase begin to assemble into mature phages. The phage nucleic acid is condensed into a compact polyhedron and it will be packaged into the head, and then the tail structures are added. This assembly of the phage components into a mature phage particle is known as maturation. From this image we can see the tail assembly occur, the head assembly occurs, and the DNA will be getting packaged into the assembled head structure, and then the head structure and the tail structure will assemble together to form the mature phage. This image shows the phage assembly of T4 phage. So the last stage in the lytic cycle is release of the mature progeny phages. It occurs by the lysis of the bacterial cell. Phage enzymes act on the bacterial cell wall, causing it to burst or lyse. And this cell lysis or cell bursting will result in the release of mature daughter phages into the environment. So, in brief, during lytic cycle, the bacteriophage attach onto the surface of its host bacteria, adsorb onto the bacterial cell surface through specific interactions, injects its nucleic acid into the bacterial cell, then the phage nucleic acid overtakes the bacterial metabolic machinery and starts synthesizing phage proteins and replicate phage nucleic acid. Then the phage proteins and nucleic acids formed will assemble together to form new phage particles, and then by lysing or breaking open the bacterial cell, the new progeny phages get released into the environment. Now we will look into a few terms related to phage reproduction, and they are eclipse phase, latent period, and burst size. So what is eclipse phase? It is the interval between the entry of the phage nucleic acid into the bacterial cell and the appearance of the first infectious phage particle inside the bacterial cell. It is known as the eclipse phase. It is the time gap between penetration of bacteriophage nucleic acid into the cell to the end of assembly. It is a time required for the synthesis of the phage components and their assembly into a mature phage particle. Now, latent period. It is the interval between the infection of a bacterial cell and the release of infectious phage particles from the bacterial cell. So, it is a time gap required from the stage of penetration to the start of lysis. It is the latent period. Now, burst size. It is the average yield of progeny phages per infected bacterial cell. It is generally 100 to 300 phages per bacteria. So, we were discussing about the lytic or virulent phages till now. They produce lysis of the host cell for their reproduction. Now, we will discuss the lysogenic or temperate phage and their life cycle. Lysogenic cycle is a mode of reproduction by lysogenic phages or temperate phages. They enter into a symbiotic relationship with their host cell without destroying the host cell. Lambda phage that infect E. coli is an example for lysogenic phage. When a temperate phage or a lysogenic phage attacks a bacterium, then lysogenic cycle occurs. Here, the phage adsorbs onto the bacterial cell surface, injects its nucleic acid into the bacterial cell, but after entry into the host cell, there will not be any phage replication. The phage replication is repressed by a repressor protein, a phage encoded protein. Instead of being replicated, the temperate phage nucleic acid gets integrated into the bacterial chromosome. The integrated phage nucleic acid is known as the prophage. The prophage becomes and behaves like an integral part of the host chromosome. We can see the prophage in this image. The phage DNA is blue colored and it got inserted into the bacterial chromosome, and this inserted phage DNA is known as the prophage. So, as the bacterium reproduces, the viral nucleic acid also gets replicated along with the bacterial chromosome and is transmitted into the daughter cells. This phenomenon is known as lysogeny. In this image, we can see a bacterium having a phage DNA in it inserted into its chromosome as a prophage. When such a bacteria divides through binary fission, there will be two bacterial cells formed, and the two daughter bacterial cells formed after binary fission, they receive the prophage along with the bacterial chromosome. Here, the viral nucleic acid is getting replicated along with the bacterial chromosome and is transmitted to the daughter cells. A bacterium that carries a prophage within its genome is known as a lysogenic bacterium or lysogen. But a bacteria cannot go on forever like a lysogen form. Under certain natural conditions or under artificial stimuli, such as exposure to certain physical and chemical agents such as UV rays, hydrogen peroxide, nitrogen mustard, etc., the prophage may become excised from the bacterial chromosome and initiates a lytic replication. This is known as the spontaneous induction of prophage. Here, the prophage become excised or get separated from the bacterial chromosome and it will initiate a lytic replication. It will take over the bacterial metabolic machinery, synthesize the phage capsid components, replicates the phage DNA and get assembled to form progeny phages. And these progeny phages will get released from the bacterial cell by causing bacterial cell lysis. So after the spontaneous induction of a prophage, the rest of the phases occur in a fashion similar to that of a lytic cycle. A lysogenic bacterium is generally resistant to reinfection by the same or related phages since it is already having the phage DNA in a prophage form inside it. So such a lysogenic bacterium is not infected by a second phage. This resistance to a reinfection by same or related phage is known as superinfection immunity. We can see lysogenic cycle in this figure. Here, a phage infects its host bacterial cell. The phage DNA becomes integrated into the host genome as a prophage, and as and when the bacterial cell divides, the prophage DNA also gets replicated along with and reach the daughter bacterial cells. This goes on until some stressful condition arises, and under the stress conditions, the prophage DNA gets excised from the bacterial chromosome and initiate the lytic cycle. During this particular period, this lytic cycle, the phage DNA will take over the bacterial metabolic machinery, synthesizes the phage proteins, replicates the phage DNA, and they get assembled to form progeny phages and get released from the bacterial cell by causing bacterial cell lysis. During lysogeny, the prophage confers certain new properties to the lysogenic bacterium. This is known as lysogenic conversion or phage conversion. So, the bacteriophages, when they are present in a prophage form, can confer certain properties to the bacterium. This type of a lysogenic conversion is seen commonly in certain clinical isolates, clinical bacterial isolates such as gram-positive and gram-negative bacteria. Their clinical isolates are shown to possess this lysogenic or phage conversion. Temperate phages are commonly found in such bacteria. They contribute to the pathogenicity in this bacteria. Examples are the toxin production in Corynebacterium diphtheriae. It is determined by the presence of a bacteriophage beta. The prophage beta form is present inside the Corynebacterium diphtheriae, which are capable of producing toxins. Similarly, Clostridium botulinum type C and type D can produce a toxin only if they are infected with phage CE beta and phage DE beta respectively. Temperate phages of Salmonella, such phage infection, their prophage form modify the antigenic properties of somatic O antigen in Salmonella. So we were discussing about the morphological types and replication of bacteriophages. Now let us look into the importance of bacteriophages. They play an important role in the transmission of genetic information from one bacterium to another, that is horizontal gene transfer by the process of transduction, and it play a role in the evolution of bacterial types and virulence. They have a role in the control of bacterial populations in natural waters. The self-cleansing and special healing properties of water from river Ganga in India is revered and known for wide centuries. And this Ganga water possesses antibacterial activity due to the presence of bacteriophages. The river water contains bacteriophages specific towards Vibrio cholerae, Escherichia coli, etc. The phages are used as cloning vectors in genetic manipulations for genetic engineering, and they are used for phage typing to discriminate between bacterial strains for taxonomic classifications of bacterial strains. The phages may be effective in treating bacterial infections, and research are now undergoing for the use of phages for treating bacterial infections, especially caused by antibiotic resistant bacteria, and this approach is known as phage therapy. So these are the importance of bacteriophages. So in this session, we discussed about bacteriophages, different morphological types, and their mode of replication. Lytic and lysogenic cycles of phage reproduction was also discussed. Thank you so much for listening.